Chapter 9, Memory. HM is a patient with epilepsy who went through surgery to remove his temporal lobes, including his amygdala, entorhinal cortex, and hippocampi. Although the surgery was successful in treating his epilepsy, he ended up with profound amnesia, a disorder of memory. He knew who he was, remembered his personal history, facts he learned in school, language, everything up until 2 years before his surgery. He could follow a conversation and remember a string of numbers for a while, but could not repeat them an hour later. So while his short-term memory was intact, he could not form new long-term memories. Since then, great care is taken to avoid removing both temporal lobes, and often a unilateral temporal lobectomy will be performed for patients suffering from epilepsy. Memory is what makes us adaptable and provides us with a survival advantage. We can avoid situations that we found dangerous in the past and seek those that were previously beneficial. Despite the vast stores of information contained in our brains, we continuously acquire new information. Learning is the process of acquiring new information, and the outcome of learning is memory. Where learning is based upon experience and represents a relatively permanent change in behavior, memory facilitates the use of prior experience. Figure 9.2 shows the hypothesized structure of human memory, diagramming the relationships among different forms of memory. Sensory memory, short-term memory, and working memory versus long-term memory. Researchers divide learning and memory into 3 major processing stages. Encoding is the processing of incoming information that creates memory traces to be stored. Acquisition refers to stimuli sustained through a sensory buffer to make a memory. Consolidation refers to changes in behavior that stabilize a memory over time, resulting in a long-term memory. Storage is the result of acquisition and consolidation and represents the permanent record of this information, whereas retrieval involves accessing stored information and using it to create a conscious representation or to execute a learned behavior, such as a motor act. The brain areas associated with memory include primarily the hippocampus and several regions connected with the hippocampus. The hippocampus is part of the limbic system and plays important roles in the consolidation of information from short-term memory to long-term memory, and in spatial memory that enables navigation. The parahippocampal region is involved in recognition memory. The entorhinal and perirhinal cortices are areas that play a key role in recognition memory. The amygdala is involved in encoding affective qualities of an experience, an example, fear. The fornix connects structures involved in memory processes. The medial temporal lobe is made up of the hippocampus and the surrounding rhinal and parahippocampal cortices. The anterior thalamic nuclei is part of the posterior medial memory system, and mammillary bodies make up part of the medial temporal lobe memory system implicated in spatial memory. Memory deficits. Amnesia refers to memory deficits that result from brain damage caused by surgery, disease, physical or psychological trauma. Anterograde amnesia refers to a loss of memory for events that occur after a lesion, an inability to learn new things. Retrograde amnesia refers to a loss of memory for events and knowledge that occurred before a lesion. Patient HM suffered from anterograde amnesia, where he could not form new long-term memories, though short-term memory was intact. He could learn new skills, but did not remember learning them. He could not recall 2 years before surgery, so along the pathways of memory formation and storage, the transfer of information from short-term to long-term was disrupted. In the figure on the left, this demonstrates coronal MRI scans of HM's brain. In the anterior slice, the hand points to where the hippocampus has been removed bilaterally. In the more posterior slice, however, the hand points to where the hippocampus is still intact in both hemispheres. This finding is in marked contrast to the belief that HM had no hippocampus, a view based on the surgeon's report that the scientific community held for 40 years. Modern reconstruction of HM's brain shows that portions of HM's posterior hippocampus were not removed during surgery. This tissue, however, shows signs of atrophy and may no longer be functioning normally. Red areas indicate where portions were removed. For HM, researchers found that memory was distinct to some degree from processes of perceptual and intellectual functions. Researchers also learned that the medial temporal lobes are necessary for forming long-term memory and for transferring information about events and facts from short-term memory into long-term memory. Also, these studies of HM suggest that the medial temporal lobes are not necessary for the formation and retrieval or short-term memories, or for learning new long-term memory that involves learning procedures or motor skills. The medial temporal lobe's memory system is involved in certain memory functions but not others, and is not critical for general intelligence, cognitive control, language, perception, or motor function. Short-term forms of memory which persist for milliseconds, seconds, or minutes include sensory memory, short-term memory, and working memory. With sensory memory, the auditory verbal information persists as an echo when you're not paying attention. If you try to retrieve it quickly enough, you find it's still there and you can repeat it out loud. For hearing, we call this echoic memory, and for vision, we call it iconic memory. The magnetic brain response known as the mismatch field, or MMF, elicited by deviant tones, shown with the blue trace, in comparison to the magnetic responses elicited by standard tones, the red traces, is shown here. The amplitude of the MMF, indicated by the shaded difference between the blue and red traces, declines as the time between the preceding standard tone and the deviant tone increases to 12 seconds. This result can be interpreted as evidence for an automatic process in auditory sensory echoic memory that has a time course on the order of approximately 10 seconds. The MMF illustrates the brain's ability to detect aberrant sensory information and respond with changes in amplitude. Sensory information enters the information processing system and is first stored in a sensory register. Items that are selected via attentional processes are then moved into short-term storage. With rehearsal, the item can move from short-term to long-term storage. In this modal model, information moves from sensory memory to short-term storage and on to long-term storage. Information can be lost at any stage due to decay and intrusions from old or new information. Studies of patients with brain damage permit a test of the hierarchically structured modal model of memory. The test here involves a sequence of 5 digits read to the participants, who were then asked to repeat the digits to the experimenter. If the digits were repeated correctly, one more digit was added to the next sequence presented. If the digits in a sequence were reported incorrectly, that sequence was repeated until the participant reported it correctly. One patient, KF, with damage to the left perisylvian cortex, displayed reduced digit span ability, about 2 items as opposed to 5 or 9 items for healthy persons. Interestingly, KF retained the ability to form certain types of new long-term memories that could last much longer than a few seconds, demonstrating a dissociation between short-term and long-term memory. Working memory represents a limited capacity store for retaining information over the short term, maintenance, and for performing mental operations, on the contents of this store, which is manipulation. Here is a simplified representation of the working memory model proposed by Baddeley and Hitch. This three-part working memory system has a central executive that controls two subordinate systems: the phonological loop, which encodes information phonologically or acoustically in working memory, and the visuospatial sketchpad, which encodes information visually in working memory. In this model, the type of information being processed is critical in determining the brain regions to be recruited. This figure demonstrates a lateral view of the left hemisphere, indicating that there is an information loop involved in phonological working memory, flowing between Brodmann area 44 and the supramarginal gyrus. And patients with lesions of the left supramarginal gyrus have deficits in phonological working memory, resulting in reduced auditory verbal memory spans. They cannot hold strings of words in working memory. Alternatively, patients with lesions in the right parieto-occipital region have difficulty with nonverbal visuospatial working memory tasks, like retaining and repeating the sequence of blocks touched by another person. Information retained for a significant time, in example days, months, or years, is referred to as long-term memory. Theorists have tended to split long-term memory into two major divisions, taking into account the observable fact that not all stored knowledge is the same. The key distinction between declarative and non-declarative memories. Declarative memory is defined as memory for events and for facts, both personal and general, that we have conscious access to and that can be verbally reported. This form of memory is sometimes referred to as explicit memory and is dependent on the medial temporal lobe. Non-declarative memory is so named because it cannot be declared, that is, verbally reported. It is also known as implicit memory, knowledge that we have no conscious access to. Several types of memory fall under this category: priming, simple learned behaviors that derive from conditioning, habituation, sensitization, and procedural memory, such as learning a motor or cognitive skill. It involves brain areas like the basal ganglia, cerebellum, the amygdala, and neocortex. Figure 9.9 shows changes in local cerebral blood flow measured with positron emission tomography. Verbal and spatial working memory tasks were tested in healthy volunteers. In each case, the views of the cortical surface show the left hemisphere, superior, and right hemisphere. Declarative memory can be further divided into episodic memory and semantic memory. Episodic memory refers to memories of personal experiences that we recall about our own lives: what, where, when, and with whom. Whereas semantic memory refers to objective knowledge that is factual in nature but does not include the context in which it was learned. Again, non-declarative memory is also known as implicit memory. Procedural memory is one form of non-declarative memory that depends on extensive and repeated experience. Tasks that require us to use procedural memory include learning motor skills like how to ride a bike, type, or swim, and learning cognitive skills such as how to read. One test of procedural learning is the serial reaction time task. In one experimental setup, participants sit at a console having 4 buttons. Participants would press buttons that correspond to locations of stimuli in front of them. Each button corresponds to one of 4 lights. The task would be to press the button with the finger that corresponds to the light that is illuminated. Over time, normal participants respond faster to the repeating sequence than they do to a totally random sequence. With amnesic patients, it was found that those with dense anterograde amnesia improved their performance for repeated sequences over a series of days. and their improvement was shown as a speeding up of reaction time. Even though they state that they have never performed the task before, these amnesic participants have learned the procedure. Therefore, procedural learning can proceed independently of the brain systems required for episodic memory. Another form of non-declarative memory is priming. Priming refers to a change in the response to a stimulus or in the ability to identify a stimulus following prior exposure to that stimulus. Priming can be perceptual, conceptual, or semantic. A word fragment task demonstrates that participants benefit from having seen the words before, even if they are not told and do not realize that the words were in the previous list. Two other domains of non-declarative memory include classical conditioning, a type of associative learning, and non-associative learning. In classical conditioning, sometimes referred to as Pavlovian conditioning, a conditioned stimulus is paired with an unconditioned stimulus and now becomes associated with it. The conditioned stimulus will then evoke a conditioned response similar to that typically evoked by the unconditioned stimulus. Russian Ivan Pavlov received a Nobel Prize after demonstrating this type of learning with dogs, which started to salivate at the sound of a bell that Pavlov rang before he gave them food. After conditioning, the bell caused salivation even in the absence of food. Two major areas in the medial temporal lobe system are important for memory mechanisms. The hippocampus is critical for the formation of long-term memory, as is with the encoding and retrieval of episodic memories. Memory recognition involves the hippocampus as well as surrounding cortices. The amygdala is known to be important in emotional memory, an example mediating the fear response. Figure 9.35 shows the model of two neocortical systems for memory-guided behavior. The components of the anterior temporal system are shown in red and are responsible for the early stages of processing. The posterior medial system is shown in blue and is responsible for later stages of memory processing. The regions joined by arrows connect together to integrate memory formation. Another fMRI study looked into testing for the involvement of the hippocampus in information encoding. Healthy volunteers were presented with words on a screen at a rate of about 1 word every 2 seconds. The words printed in either red or green were of either animate or inanimate items that were either large or small. Depending on the color, the participants were required to make either an animacy judgment or a size judgment for each item. While the participants viewed the words and made their decisions, they were scanned using fMRI. Later, outside of the scanner, they were tested on their memory for the items that had been shown by being presented with the 360 old items that were mixed with 360 new items. Participants were asked to rate on a scale of 1 to 6 how confident they were that they'd seen the word before, and specify whether it was in green or red. It was found that correctly recollected words activated regions of the hippocampus and posterior parahippocampal cortex during encoding. At encoding, participants viewed a series of words and made either an animacy, animate versus non-animate, or size, large versus small, judgment for each word, depending on the color of that word. Later in a test at retrieval after the scan session, participants made 2 decisions about the items presented, which included the old items and new items never seen before. First, participants were asked to indicate whether and how confidently they recognized the items. Second, for each word, they had to make a source memory judgment. Had it previously been presented in red or green? Figure 9.26 shows horizontal sections through the brain with the hippocampal area circled in red. The figures below demonstrate hemodynamic responses from event-related fMRI measures taken during the retrieval of previously studied words. The hippocampus was activated by correctly recollected words, as shown by the solid red line, but not by words that the participants had previously seen but could not recollect, indicating that the words merely seemed familiar, the solid black line. The hippocampus was activated for episodic recollections, not for memory that did not contain awareness of the prior event. That is, when items recollected merely seemed familiar to the participants and were recognized by their familiarity alone. Figure 9.7 presents the neuroimaging results from analysis on familiarity, testing participants on their confidence in whether they had seen an item before. It is shown that brain activity during encoding correlates with the confidence of recognizing that an item has not been seen before. Functional MRI activations that correlated with confidence ratings can be seen in the entorhinal cortex. The graph shows that as recognition confidence increases, activity in the pentorhinal cortex also increases. Images of the left and right hemispheres show additional regions of cortical activation. Alzheimer's disease is a neurodegenerative disease characterized by dementia involving a loss of memory and other cognitive abilities. It largely affects the hippocampus brain region and is characterized by amyloid plaques and neurofibrillary tangles in the hippocampus. These diagrams depict a normal section of cortex with cortical neurons and a section of cortex in an Alzheimer's patient containing amyloid plaques between neurons and neurofibrillary tangles within neurons. Beta amyloid plaques are sticky clumps of protein fragments and cellular material that form outside and around neurons. Neurofibrillary tangles are insoluble, twisted fibers composed largely of the protein tau that build up inside nerve cells. These plaques and tangles interfere with normal hippocampal cell function and cause disturbances in memory mechanisms. As indicated, the cellular basis for memory involves hippocampal mechanisms. One mechanism, long-term potentiation, is a pattern of electrophysiological activity that relies on NMDA receptor function and is an indication of synaptic strength. It can be studied in hippocampal brain slices from mice and rats with electrophysiological techniques. NMDA or N-methyl D-aspartate receptors are critical to inducing LTP, but not maintaining LTP. LTP does not occur unless the neurotransmitter glutamate is present in the synapse to bind to postsynaptic NMDA receptors. Activation of the NMDA receptor allows sodium and calcium ions to flow into the nerve cell. When the channel is blocked by magnesium ions, it normally prevents the calcium channel of the NMDA receptor from opening in response to only glutamate. Associative long-term potentiation reflects increased calcium entry produced by changes in NMDA receptors. The notion that learning a conditioned emotional response to a tone involves long-term potentiation is supported by studies in which a drug that blocks the NR2B subunit of the NMDA receptor also blocks learning.